Hello! Today I'm going to give you a tour through the animal cell. First off, we are going to start at the cell membrane, which is outside the cell. The cell membrane is a thin and flexible barrier that surrounds the cytoplasm, made up of amphipathic lipids that regulate what enters and leaves the cell via a passive and active transport mechanism. Specific proteins are embedded in the cell membrane and can act as molecular signals that allow cells to communicate with each other. The cell membrane also has carbohydrates attached to the lipids to help keep the cell membrane together, along with the proteins. Now moving on to the brain of the cell, the nucleus. The nucleus is an organelle that is the information processing center of the cell. The nucleus has two major functions, which are to store the cell's DNA and to coordinate the cell's activities, which include growth, intermediary metabolism, protein synthesis, and reproduction. The nucleus is the largest organelle in the animal cell, and it has holes called nuclear pores that allow specific materials to pass in and out of the nucleus, just like proteins in the cell membrane that regulate the movement of molecules in and out of the cell itself. Now moving on to the nucleolus. The nucleolus is the nuclear subdomain that assembles ribosomal subunits in eukaryotic cells, and its primary function is to assemble ribosomes. The nucleolus organizer regions of chromosomes contain the genes for pre-rRNA, which is the foundation for the nucleolus. The nucleolus disassembles at the beginning of mitosis, while its components disperse in various parts of the cell, and reassembly occurs during telophase. Now moving on to the cytoplasm. The cytoplasm is a jelly-like material that is mostly made up of water and fills up the cell. It contains proteins, molecules, and important substances that the cell needs. The fluid part of the cytoplasm is made up of proteins, lipids, carbohydrates, salts, sugars, amino acids, and nucleotides. The cytoplasm contains dissolved nutrients and helps dissolve waste products. The cytoplasm helps materials move around the cell by moving and churning through cytoplasm streaming. The cytoplasm also works with the nucleus by taking the waste from the nucleus by synthesizing DNA. Next up we have the centrioles. The centriole is a barrel-shaped organelle found in most eukaryotic cells. The walls of each centriole are usually composed of 9 triplets of microtubules. In replication, each new paired set of centrioles are composed of the original centriole, plus a newly made centriole. If the centrioles are used in forming vesicles and also motility organelles, flagella or cilia, the older of the two centriole, the mother centriole, becomes the basal body which organizes the structure of the organelle. Now to the chromatin. The chromatin is the structure that the eukaryotic genome is packaged into, allowing over a meter of DNA to fit into the small volume of the nucleus. It is composed of DNA and proteins, most of which are histones. The DNA-protein complex is the template for a number of essential cell processes, including transcription and replication. These processes depend on the precise structure of the chromatin fibers in order to take place. Microtubules. Microtubules are the third principal component of the cytoskeleton. They are rigid hollow rods that are approximately 25 nanometers in diameter. Like actin filaments, microtubules are dynamic structures that undergo continual assembly and disassembly within the cell. They function both to determine cell shape and in a variety of cell movements, including some forms of cell locomotion, the intracellular transport of organelles, and the separation of chromosomes during mitosis. Microfilaments. Microfilaments are solid rods made of globular proteins called actin and are common to all eukaryotic cells. Long chains of the molecules are intertwined in a helix to form individual microfilaments. These filaments are primarily structural in function and are an important component of the cytoskeleton, along with microtubules. Ribosomes. Ribosomes are tiny organelles composed of approximately 60% ribosomal RNA and 40% protein. Although they are generally described as organelles, ribosomes are not bound by a membrane and are much smaller than other organelles. Ribosomes are mainly found bound to the endoplasmic reticulum and the nuclear envelope, as well as freely scattered throughout the cytoplasm, depending upon whether the cell is plant, animal, or bacteria. Ribosomes serve as the protein production machinery for the cell and are consequently most abundant in cells that are active in protein synthesis. Rough ER. Rough ER is a system of interconnected, membranous, enfolded and convoluted sacs that are located in the cell's cytoplasm. Rough ER is covered with ribosomes that give it a rough appearance. Rough ER transports materials through the cell and produces proteins in sacs called cisternae, which are sent to the Golgi body, or inserted into the cell membrane. Smooth ER. Smooth ER is a vast system of interconnected, membranous, enfolded and convoluted tubes that are located in the cell's cytoplasm also. This space within the ER is called the ER lumen. Smooth ER transports materials through the cell. It contains enzymes and produces and digests lipids and membrane proteins. Smooth ER buds off from rough ER, moving the newly made proteins and lipids to the Golgi body, lysosomes, and membranes. The Golgi apparatus. The Golgi apparatus is a network of stacked membranous vesicles present in most living cells that functions in the formation of secretions within the cell. The Golgi apparatus is made up of smooth cisternae, which are coated with lipid membranes. Its main function is to direct molecular traffic in the cell. Lysosomes. The main function of lysosomes are to serve as digestion compartments for cellular materials that have exceeded their lifetime or are otherwise no longer useful. Lysosomes recycle the cell's organic material in a process known as autophagy. Lysosomes break down cellular waste products, fats, carbohydrates, proteins, and other macromolecules into simple compounds, which are then transferred back into the cytoplasm as new cell-building materials. To accomplish the tasks associated with digestion, the lysosomes utilize about 40 different types of hydrolytic enzymes, all of which are manufactured in the endoplasmic reticulum and modified in the Golgi apparatus. Lysosomes are often budded from the membrane of the Golgi apparatus, but in some cases they develop gradually from late endosomes, which are vesicles that carry materials brought into the cell by a process known as endocytosis. Vacuoles. Vacuoles are fluid-filled, membrane-surrounded cavities inside a cell. The vacuole gets filled with food being digested and waste material that is on its way out of the cell. Vesicles. Vesicles are small, membrane-enclosed sacs which store and transport substances to and from one cell to another and from one part of a cell to another. The vesicle is separated from the rest of the cytoplasm by at least one phospholipid bilayer. Vesicles can fuse with the plasma membrane when they want to release their contents outside the boundaries of the cell. However, vesicles can also fuse with other organelles that are present inside the cell to release or engulf substances. Mitochondria. The mitochondria turns sugar into energy for the cell to use throughout its functions. Mitochondria provides the energy a cell needs to move, divide, and produce secretory products. Pretty much, they are the power centers of the cell. They are about the size of bacteria but may have different shapes depending on the cell type. Flagella. The function of the flagella is usually to propel a unicellular or small multicellular organism by beating with a whip-like motion. In larger animals, the flagella are often arranged en masse at the surface of a stationary cell anchored within an organ and serve to move fluids along mucous membranes, such as the lining of the trachea. The cell theory states that all living organisms have a basic unit of structure and function, which is the cell. This was a biologically significant statement because it suggested that all living things have a common denominator. Almost 200 years of research by many different scientists led to this conclusion. The initial discovery of cells was done by Robert Hooke in 1665 when he designed one of the first microscopes. He looked at some cork through the microscope and saw many units he then named cells. Hooke and other scientists observed other samples of plant material and discovered that they were also made up of cells. As they began examining more material, they noticed a pattern. It wasn't until 1838 that a German scientist called Matthias Schleiden stated that all plant material was made up of cells. The following year, Theodor Schwann came to the same conclusion about animals. Their findings are what have become known as the cell theory. It's time to go. Hope you had fun. See you next time. Bye!